Yes. Oh, okay. We already live on YouTube. Did you see there? Meeting is now streaming live on YouTube.
Yes.
Okay. Well, can I start right now, Professor Heike?
Yes, I'm happy.
Okay. Well, audience, we will continue our webinar about sequence and sequence analysis of marine organisms. It will be held, or our speaker is Professor Heike Wagele that from ZFMK. Now I give the time to Professor Heike.
Good.
Please.
Yeah. Thank you very much, Billy. And thank you to all of you that you want to know more about sequence analysis. I think I will just share the screen now so that we can start. I hope you can see this now. Just Billy or Mehdi, can you see the screen?
Yes, we can see.
Oh, very good. Okay, so those who have also attended the last two lectures, you should then have in mind a little bit about especially how sequences can be used, in which form they can be used, how to retrieve sequences. You can retrieve the sequences by yourself, from material that you have collected, but there's also a lot of information published in public databases or repositories. And these data are also available for everyone on the world. And on these repositories, these sequences you should actually also use when you analyze your own sequences. So you never should analyze only your sequence and say something about only your sequence. You usually have, well, you always actually have to analyze your sequence in combination with published data. That means with sequences from these repositories. Then, so you got all the sequences from NCBI, you got your own sequences, and now we have to talk about how the alignment is actually done, because the alignment is the most important part for all subsequent analysis. And I said a little bit already about final alignments. When you use programs we have talked about, like BioEdit or Mega, that these programs, they can cause problems when you use the algorithms of these programs for doing an alignment. So there are many other alignment programs that can be used and that are free available, but all of them have some pitfalls too. And maybe another pitfall that can be also seen in the other alignment programs. So why is it so important to use special algorithms to get your final alignments? This is because they are using different algorithms. I am not an informatician, mathematic person, therefore I don't know what this actually means, which kind of algorithms. For us, we use these programs like a black box. We put the data in there and then something comes out, and usually a lot of people believe what it comes out and they don't dare to change anything. But this is not true, as you will see later on, and you can also manipulate a little bit. There are, as I said, several free available algorithms or programs, and they are used for aligning the nucleotides or amino acids. For example, Clustal W. This is the one that is implemented in BioEdit and Mega, and also in Genius. Genius is a program that is actually very, very good, but you have to pay for it, whereas BioEdit and Mega are free software. That was developed in 1994. If you want to know more about it, actually you can go to Wikipedia and look at these, how they are working. So this is a progressive alignment. how it works, I'm not really quite so sure, but usually they take two sequences, align them to each other, make a consensus sequence, and then they take the next one, align it again, and so on. Then we have muscle. Muscle is already much better. It is based on a similar algorithm, but then it has iterative alignments. That means it's running the analysis again and again and twice to make your alignment better. So this is 10 years later. It's also implemented in Mega. And MAFT is actually the one that is used most now and is the best at the moment, has also a lot of possibilities. I will show you later on. This was developed in 2005. It's a download version or an online version. I usually use the online version, and that's what I also will show you today. So when you open MAFT, just to show you a little bit how it is working, I've shown you already how to put the sequences into one file. So you have all your sequences in your file. Now you want to work with this file and do the alignment. This is when you open it online here. It's called then the server. And then you see this page. You actually, for the beginning, you don't have to use anything of this one. You just start here, the input. You have several options. You can actually include here the sequences directly, like you see these in editor. Or you say here, sorry, this is in German now, search. So you can search for the file that you have created, that we have actually created already last week. Next thing that you can, so for example, we are taking, of course, I will already show an example. I use the Commodores alignment, the Commodores, how did I call it? The amalgamation of this, all the sequences that we put into one file. We call it Commodores. You see here 16S, I use the 16S file. And then we have here already the first options to choose. I'm not going into this one. This is actually what you don't need. The uppercase, lowercase is just how the output looks like. And it's better to use this one in case you have used this one because, whatever reason, you still can change it. There are programs you can change it. Of course, that's also the default, same as input. This is default, this is default, same as input. You can leave it as it is. And the output order, of course, you want to have this aligned because you want to have an alignment. Then you have the job name. Give it a job name because this name then appears in the email that you get. It appears on when you get the results. It helps you to keep a little bit sorted out, because when you're doing several analyses in parallel, it's good to know which one you open then. Okay, so we talked about this output order. It's aligned. We have here the job name here now. Then you put your email address in there. You don't have to. You can also just stare on the screen and see what happens. But when it takes a little bit longer, maybe you do something else. So it's always good to put your email in there. Then you get the results also per email. Now you can actually already submit, but I want to explain you also a little bit what comes later on, because when you're working a lot with MAFT, when you're working a lot with different kinds of sequences, then it might be of interest to also see what else is available. So per default, we have this auto, and you can leave it as it is, then submit, and it's fine. But there are also other methods, other algorithms that can be used. For example, when you have more than 2,000 sequences, I've never done that. Actually, the largest that we had with Adel was about 800. So 2,000, then of course the program takes a lot of time. There are algorithms that can speed up, but usually then this is not so accurate. Then there's another one. Don't ask me what this is behind there. I can't tell you. This is another one that is fast, and it's a progressive method that is then already more accurately than this one here. This one is actually a very slow one. And for this, you need to have actually already a tree. And then it can align again along a tree. That means the tree already helps to show which of the sequences should be more similar. Now coming here to below, this is then the next step that is even more, taking more time. So this is the iterative refinement method. That means it starts with a progressive method and then goes into the refinement. And this is then much, much slower, of course. So we have here, for example, this is slow, and then you see very slow, very slow. And this is actually only when you have much less sequences than 200, 300. When you have more than 200 sequences, this is not working then anymore. Yeah, you wait for days or even longer. So this, when you have not so many sequences and when you have multiple conserved domains, so this is then mainly actually for ribosomal genes, for example, and when you also have long gaps. You might have long gaps when you're working with several genes and you do not have information for each of the specimens you are working with. Then you have lots of gaps in that. You will see this later on. Then this might be good. But it has only 2 iterative cycles. And sorry. And when you go for the other ones, then you have even more possibilities, as you can see. But usually with what you are doing with the CO116S, you can just go for the auto. Okay, so now I was sticking too long to this slide. Okay. So if you further go on there, down in this program, you have even more options. So for example, you see here something, this is really wrong. And then you can provide an information to that these things that do not match at all to the other stuff, to put this in gaps. You don't need this actually. This one here down is more mainly for the proteins, if you're working with proteins. Here is with the guide tree. That's what I just said, when it has when it has to be very, very accurate. But this is the default situation. You don't have to use change here anything. So this MAFT homologs is new for me too. I didn't work with this in the past. You can just forget it. This is not really a big issue. And the same is also here, the plot last hits. That means the top sequences versus the others. That means also that this top sequence is what we will see later on, actually your ouch group. You can just leave it as it is, and then you can submit. So when you submit, you get at once this file that says submitted. It said then notifications will be sent to, it says Wagele, so that's my address. It says the job, Commodores, and then you see here the plot. The plot tells you about the quality of your data you have given in there, and then where the alignment actually will be available. So, and this is really then the important one. And you can also, of course, cancel if you want, but usually you don't want to cancel this, except you saw that you made a mistake by not including something, and then you can stop this. Yeah, and then you can actually open this when you finally click on this and it has finished, then it opens like this. You see that here, this is the headlines actually. It says already in which format you want to have this alignment. And you remember that we were working mainly with a faster format in the last week and not in the other one. So this is actually a MAFT, the result. But if you want to have this as a faster format, you have to click then later on. There is a program implemented here in MAFT when you have to reformat it, because there are other formats that are needed for other programs. At the moment, we don't need this. Phylip is sometimes used, for example, in splitstree. MSF is an older one. I don't know where this is used at all. Nexus is a very common format that is used also for tree reconstruction, also splitstree. You can also have changed this, because you see this is now lowercase, and usually for better visualization, uppercase is better. And then if you want to change anything, you can do this with RedSec. RedSec is a program that you can use. So it says here cluster format alignment by MAFT, and you see here, perhaps you remember, this was the accession number from NCBI. And what you also see here is now there is no further information at the moment about the sequences itself. So you don't see which specimen, which species it is. It's only that you have here a long, it starts actually with the nucleotides, and we have here, that's also what you can see, gaps. There are 2 gaps in these first sequences. So what I just said, we usually download things. So we would click now on faster format. And sorry, this is German again, because I'm working on a computer that is, of course, meant to be in German with the issues. So you open. You can do this on a regular basis with editor. I have it with editor because then I see at once if something is wrong. But you could also implement here by BioEdit or another program. Then you click on OK. Or you say also, you can also say first save it and then or open it. And then you see that this MK999994178, you see it here. It has the typical FASTA format with this signal here, with this sign. And then you can see again what this actually is. It's Commodore Selena. You see the voucher. So this is the specimen here. It's 16S ribosomal RNA. And it's the partial sequence, not the full one. And it's mitochondrial, it's not nucleus. And here's already the next one, 77. Yeah, that's this one. As I said, you can, when there's something wrong, you want to change it, you can also do this with WetSec. So you say reformat. You reformat, then this opens, and then you have quite many options here actually. Sometimes it might be of interest really to do this working with different programs that I will not present here. This means, well, it starts with pretty, whatever this is, I don't know. And then you can click on it. And when you click on it, then you have many, many different forms, and including EMBL, for example. EMBL is the version how to upload in NCBI. I have told you already, when you publish your sequences, you have to upload them to public repositories. They are actually not your private property anymore. They should be made really public and available for others, and that would be a format. But we, at the moment, we want to have faster. So if it's not faster, maybe you did cluster or whatever, then you can click on this faster, and then you have the faster file. Yeah, and then it looks like this. Yeah, so we have again these sequences here below that. This is now with the editor. Yeah, I wanted to tell you, if you have this problem now, I changed it into, that's what I did. I changed also the letters into uppercase because I prefer this for showing and working with. What you can do instead, I made this mistake just to tell you, because this is the default situation. And if you don't want this, you have to click manually on this, same as input. So then you keep your capital letter. So better click directly on this icon. This is not default. Yeah, and then when you click on the icon, it comes up like this. And when you don't click on the icon, it comes up like this. So what I want to show you here is that the alignment programs really work differently. Also, this is not really difficult. This you could even do by hand, by eye. You don't need a program for this. But nevertheless, there is some issues here. And I want to show you the difference just to show you that this is true, that programs work differently, and some are better and others are less good. This is the one that I said we were doing with Clustal W. This was implemented in BioEdit. That's the one we did actually last week, quick and dirty, as I said. And I want to show you here that with these Selena, we have one gap here. And then we have here where these two gaps are, we have A and C. So later on, when we reconstruct the tree reconstruction uses the steps, the amount of mutations that it finds in the alignment. And in this case, these 8, how many? Yes, these 8 taxa, these 8 sequences, they differ in 2 mutations from the next ones. Yeah, because we have an A and a C. And this one has 2 gaps, but it has to use a gap here because there is no T in these ones here. So there's a gap here. That means in the tree reconstruction, so the next step we are doing next week, it would count these taxa with actually 2 mutations and 1 gap. And mutations, of course, are much more valuable than gaps. They count much more. When we look now in the MAFT situation, it has aligned the situation like this. It has moved these ACAs of that to that place over there. So there's 1 mutation actually here, and we have here 2 gaps. So it counts this differently in the tree later on. This is not a big issue. So the tree will not be different from that one, but it sometimes can really count how many mutations are you having in this situation and how many gaps you have in this situation. So that's why you also should always look at the alignment and see, are there any issues in that? Yeah, this is now, so you have already an alignment of 1 gene. You have seen this is very easy. When you have gathered already the assembly of all your own sequences, including the NCBI sequences, then you only use the program. And hopefully, yeah, we had already yesterday some issues with a really large data set, but in a smaller data set, you usually should not have any problems. And then you have already an alignment. When you're working, even in barcoding, you usually use more than 1 gene. Still, a lot of people use only CO1, but very often we combine 2 genes because each gene evolves in a different speed. So using CO1, the speed of mutation, the mutation rate is higher than in 16S. And this, when we want to identify species for barcoding, and very often also want to say a little bit at least about how the genera are related and in which genus it is good to have the 16S. 16S shows less mutations. So what we then usually do is we combine 2 or more genes in 1 alignment. So we combine CO1, 16S. And in this case, what I want to show you now in the next steps is we combine CO1 and 16S as the mitochondrial genes plus a nucleus gene, and in this case histone 3. And it's only a partial one. We are not using the full one. Same as here, we are not using the full one. So you have then more nucleotides in your as information. You might remember that CO1, we usually have about 600 to 650, that's normal length. In 16S, the normal length for this partial one with the primers that we are using and that are available worldwide, we usually have then about 300 to 400 base pairs. And the histone 3 would add additionally about 300 to 400 base pairs. So then you have an alignment that is longer than 1,000, about 1,500 base pairs. And that is, of course, much more information. So working now with 3 different genes, and I want to use a new project. This is Tambia. This is another C-slug, of course, and I want to go through with step by step with you so that you remember how we did it last week. You remember that I said if we want to work now on Tambia, on these 3 different genes, we have to go into NCBI first because we have to download, of course, the what is available in the repository. So we call it Tambia. I put this in organism, not to get any bacteria, and yeah, we don't want this. And then we put 16S in there. So we search for this, we click on that, and then we see we have 17 items that it was found. Just a quick check. This is Tambia marbayensis. This is Tambia gabriele. It's always 16S, 16S, 16S. This is fine. Well, we asked for 16S. So you click then on send to. It comes up with complete record. You choose the destination to the file. Please choose here again faster. Yeah, the default situation is different. Then you click on create file and you have this. when you automatically open FASTA files with editor, you have this. When you tell your computer to automatically open it in BioEdit, then it will open in BioEdit. But as I said, I prefer this with the editor, and then I can see whether I have all information here already. So we have here the accession number of NCBI again, and we have the names and 16S, everything, the information we need is there. Next step is cytochrome. We want to have CO1. And CO1 is a problem because there are different names for that. I think I mentioned this already last time. You can use COX-1, you can use it like this. You can use, say, cytochrome. You can use also this COI, and you get different results. So COX-1 did not work in this case. CO1 did not work. When I put the name cytochrome in there, I got 38 issues, as you can see here. And when I only use CO1, I get only 31 sequences. So you see there are differences. In this case, for example, when we just go up here, look at this. Yeah, I did the same again, send to create file and so. I got the editor opened. And then you see this is only cytochrome oxidase subunit 1 gene. There is nothing like COX-1. There's nothing like this mentioned, nothing like this. So if you would include something like this, you would not get this sequence. You would not get this sequence. Only with the cytochrome. Only the cytochrome worked in this case. When you, I only use now here the cytochrome. Of course, then you also get those ones where you have the word cytochrome. And here we also have CO1. So interesting is now that although these are the same people who use, who uploaded the sequences, in NCBI we have different, the mentioning of the genes is a little bit different. And you have to be aware when you're working in NCBI that there might be different names for the gene that you want to use. So 16S, for example, can also mentioned be as large subunit mitochondria. Not even 16S, might also be just large subunit. And you have to be aware, and you have to make a little bit more search to get really all sequences you want. Yesterday we made a quick search on another group, not on the C-slugs, and I also only included cytochrome. And then you might not only get a cytochrome CO1, the CO1 part, you might also get cytochrome B. And cytochrome B is a completely different gene area, and you have to be careful because these, of course, you have to erase. So you always have to check whether you are really working here with the same gene and not with different genes, and they have just a part of the name is the same. Yeah. So cytochrome oxidase subunit 2 is not cytochrome oxidase subunit 1. So you have to be careful here. And now we had the two mitochondrial genes we have downloaded from NCBI, now histone 3 or H3. So it's as you can see here already, I made the search already with Tambia, organism, and histone. So it says here histone and also H3, but sometimes it's only histone. So you have also to be careful. There are other nuclear genes. We were talking about 18S and 28S. I was looking for them just to see, has anyone worked on 18S and 28S in Tambia? And I had no hit. So we have to stick to histone if we want to have a nuclear gene in there too. So now working on the sequences in the sequence editor, so in the program BioEdit. As I explained this a little bit already last time, this is in a sequence alignment editor. And what we have to, and you have to transform, of course, the text files when you worked with an editor, you have to transform the files then in a proper FASTA file, because the FASTA file, and that's, I told you already last time, this is, the FASTA file is recognized by many programs. So here, for example, in this case, I saved them all as an editor in this editing, the text file. I have here Tambia 16S, I have Tambia 01, Tambia H3, but then of course you can also then click on save as here. I have all files, but you can also click on this and save as FASTA, and then you have them really as FASTA files. But sorry to say, BioEdit reads these text files, yeah? So you go into BioEdit, you open BioEdit, you click on this text file, it will open this Tambia 16S, and you will see the sequences here, but then please save them as FASTA again. So we have here the Tambia 16S file, Tambia, Abdere, Affinis, and so on. You see it's the 16S, and you see it's not properly aligned. You see that there are similarities here, but we do have to do an alignment. Yeah, what we can also do here is now, if you want to change something, because we have here the accession numbers, and working with the accession numbers does not tell you anything here about the species or the genus you are working with. Nevertheless, this is now very important. You have to keep somewhere these accession numbers, because you will see when we concatenate, when we combine several genes, we cannot use these accession numbers anymore, because the accession numbers change for the different sequences. Although we sometimes should have the same specimen, the same voucher number, there's a different accession number for the different genes. And so, but later on in the publication, you have to know these accession numbers because you have to mention them. If you want to change anything in these headings, what you see here, you can click on it. I highlighted this now in black. Well, it is black when you click on it, and then this opens. And here you can change whatever you want here. You can even change here these the nucleotides later on when you see there might be still some mistakes in there. So this is a very good issue in BioEdit where you can change things. What you also can do is you can include many more informations, for example, for your own sequences if you want. And this then also helps you later on to remember where did you get the specimen, who gave it to you, or where did you retrieve it, and so on. So you have in BioEdit the possibility to put many more metadata in this information here, and also to change these metadata. What you can also then do is you have, now we have only the NCBI data in there. Of course, we also want to have our own data in there. And this is by, if you click on File, Import, Sequence Alignment File, then your usually Explorer opens again. So I will do this now. And then, of course, you are in your Explorer again, and then you can choose whatever you want to include. Of course, remember that we are working here with the 16S, so don't include any CO1 or H3. You have to include some other sequences, maybe your own sequences. I made a file, I called it own fake sequences. So these are 16S sequences, and I want to include them now here. So you click on them. It's still, again, it is a text file. It doesn't matter. You click on it, you open it actually, and then this is actually the fake, I call it own fake sequences. I put a Polycera in there. So you see it's a different genus. Polycera, there's a Tambia that I found that is not identified yet. So I did not make these sequences, I made them actually up. But that's the ones that we want to include now in this file. So we just open it, and then it should end up here. And you see we have here the Polycera hedgepeti, Polycera atra, Alaba, Tambia spec, and the Polycera ferroensis. And you see, all right, these two, they look already aligned. Yeah, so they start from the same place, and the others are not aligned at all. But what is also now very important here, and this is you have also to keep in mind, all the programs we are using later on will use the information that is connected in some way. So in this case, Polycera atra, I opened this window again, you see Polycera atra is not connected. So for example, the phylogenetic programs would only use Polycera, and the atra they would not recognize as part for the identification. And that means you have here Polycera, and the same is here with this Polycera. When there's no line in between, it will not recognize this as Polycera alaba. You would have two Polycera sequences and you would not know later on which one is what. So what you have to do is, and that's also that you have to check actually, that you include this underlining, and so connecting Polycera with the species ending atra. This is really important. And when you use, for example, this here, you can also see it very quickly whether you have it underlined or not. So, okay, and now you see already we have so many alignments. We have started already with CO1, we started with 16S, we have a histone 3, we have the NCBI file, we have your own files from your own sequences. You really have to think about good names. I always also make sometimes mistakes, but it is really good to include the genus you're working with or the family that you're working with, and always also the gene, so that you not mix up later on the genes you are working on. So in this case, I put the name in there, Tambia, that NCBI means all the sequences from NCBI, and down there, the fake are my own sequences, and it's 16S. And remember, click down here on faster and then save it. So this is still only an assembly. This is not an alignment. Now we're going to align it. You remember, we were talking about this already. You click on this search. You click on the file you want to do now the alignment. Tambia NCBI fake 16S, we call it. You click on that, don't forget it, same as input. You put your job name there in there, Tambia. And you put your email address down there. Yeah, so we have here now Tambia in there. You put your email address in there. And so we use here the default settings. We do not forget the email address, and then we just submit. We keep the strategy, the default strategy auto, so we don't care about the other things. This is not a complicated alignment. Now we put the email in there, and this is now my email address. It starts with MAFT online alignment job started. Then it tells you alignment later on. You see this was very quick. It was very easy, and the server, nearly nobody was working on the server. So you see here the job has finished, and then you also have a plot. You can look at this, but this is not so important. So this is actually what I opened here. The alignment Tambia has finished. You can click on this, and you can also click. This is something that might happen, to check the direction of the nucleotide sequences. We had the problem yesterday, but very often this is not the case. So you open that, and then you have, again, this alignment that looks for me very nicely. We have all the sequences in here. And you see here also that this is one specimen, and this is the sequence coming below. And it does not end here. It goes, it starts again here, and it will go on until it is finished. So it's not that we have different specimens here. This is actually a block that is repeated down here. So I set faster format. It opens with the editor. We have it here. So that's actually the same as we see here. But we also see already the full, oops, where is it? Yeah, this was not so good. No, here it is. I'm sorry. So we have here the full CO1, 16S. Here starts the next one with Tambia affinis. So save again under a good name. What I usually do is, so this is NCBI now, fake, and Ali. Ali I use usually for a line. That helps me then to find the file again where I have the aligned sequences. So you save this under good name, Tambia, 16S, NF, Ali, and then you can open it with BioEdit. Or you have opened it already directly with BioEdit. Yeah, and here it is. This is the original one we included. And this is now the new one. And you see here, this was not aligned properly, but now it's really aligned very nicely. And then you also see that here, for example, this sequence, there are a lot of gaps in there. So in this case, it's the capensis. Where is the capensis here? It's here. You see, it looks so different here. It's because it also starts, of course, in number 1 here, but now it is properly aligned and now it starts actually here. So you see, when we are looking here, the position, the homology of this position, the T, that's it's violated. It's not true. But now here it's correct. The T belongs to this. This is our, actually, this is the homology hypothesis. You also see that this is really different, but you see the pattern that this differs from this one. But when you go on, don't only look at this. When you go on, it's really properly, nicely aligned. So this is correct. These are just differences, genetic differences of these two. That's Abdere. You can see Thambia Abdere differs in several positions from the other ones. Whereas here, Gabriele, Morosa, they are much more similar. So Abdere, I see it at once, it is different. Here, this one is also quite different, but this is a different genus. So I would expect it to be different. There should be more differences, hopefully, yeah, later on. Now, because we are talking, this is the 16S. And you remember that 16S is the ribosomal gene that has conservative areas like this. This is really conservative. These are the ones that are actually matching again. Yeah, so we have to this part a counterpart that matches the complement. And whereas these areas that can vary a lot are the loop areas. So when we move further on in the alignment, now we are talking about positions 200 to 370. We see here again, this is very conservative. This is where they are binding, the binding areas, the conservative areas. And here are a lot of gaps, but we have information here. So we have a loop area in the Polyptera species, but the loop area in the Thambia specimens is much, much smaller. And same here and same here. So this is what tells me this is the area, the loop areas of the sequence. So that was the 16S. Now we do the same procedure for Thambia. We downloaded already the NCBI sequences. This is now before we aligned with MAFT, but we can include, of course, next our own sequences, what we call now the fake sequences. With the CO1, I had now only 3. I do not have all of them, so I used the Polyptera, 2 Polyptera species and Thambia species again, and it's aligned in MAFT, and then it looks like this. Yeah, so I showed up here a little bit, and you see that the Polyptera, they differ in CO1. Yeah, that's quite different here, and we also have a long gap here. There is this one, for example, has much more information here in the beginning, but most of this is actually starting here. So the next step, what we would do here in this case is also check the whole alignment. Is this okay? We would go through it. Are there any queries? And then we would also trim. So you see, this needs to be trimming because in the phylogenetic program, this would be used as an information, and it would actually tell that this sequence is special because it has so many things, so many nucleotides, all the others do not have. And that's actually not true. This has something to do with how the raw sequences are obtained. And the primers, maybe the people who uploaded it, cut off their sequence somewhere here because the primer are not working properly in the other region. And the person who has uploaded this one, maybe it even included the primers, I don't know, but we would cut off this. If we go here for the back, yeah, so I moved to the back to this CO1 sequences. It's a very long one, 690 base pairs. And again, we have here the same one, it's extremely long again. So we have to cut this off and also this one because this is no information. But for the rest, that's fine. So we would cut off it actually at 696. And that's what I do. Yeah, BioEdit has its pitfalls. If you have problems in doing this, just close this the file again and open it, and then hopefully it works. If you want to get rid of this this part, you actually mark it. You go from here to the beginning. You have to click edit, and then you can delete it. When it does not work, maybe you have not clicked on edit, you have select or grab and drag, and then it does not work. Sometimes you have to close this. This is actually open for more manipulations. Sometimes you have to close it and open it again. Then it works. Or as I said, just close the whole file and open it again. So we deleted this one. Sometimes it is also quite good to use this function, especially when you have a whole genome in there. You can go, you click on one of these, actually on this position, and then this position will be marked. And then you go to edit, select to end, and then the rest until the end of the alignment is selected, and then you can delete it. It happens that you might have a whole genome in there. The whole genome, then you are not ending at 700 or 800, you end at about 15,000 and something. And you don't want to go through manually from here to 15,000. So you would select to end. And then you delete it again, and then you get rid of this. It's also good to know about another function in BioEdit that helps you also to see the variability. So you have these different buttons. This I like to, well, you can see it already here. You see that I use that the whole tile is actually colored. And for this, you have usually to click on this one. Per default, it comes like this. I don't like that one, but you can just play around. And this one is quite nice to see actually where you have differences and the same nucleotide on a certain position. So we see here, we have a T throughout the whole, all sequences. But here, for example, we have a C and a T, C, T. And because this is a protein coding sequence, you can already check whether there are really still mistakes in there. A protein coding alignment should always be like this. One position, the first and second position are quite conservative, and the third position of the triplets for the amino acids can vary a little bit more. It's not always working like this, but usually yes. So for example, we have 2 conservative, nothing has changed. The third position, there is variability in there. Next, first, second column are quite conservative. Third is variable again. But as I said, it's not always working like this, but it gives you at least a little bit of information of this. Okay, so we have the CO1 now. Now we are going to histone 3. We downloaded already from NCBI. I included here already our own sequences from histone 3 that I could find. It's already, oh no, it's not aligned yet. I just put them together. And now, and what you see here already is it's very conservative. There's not so many changes. And I already see from the color that these nuclear genes, they have very often also a bias towards more certain nucleotides. So in this case, the cytosine is very, very common. It's in a high percentage, but this is not unusual. Okay, so this was already aligned. Yeah, it is already aligned. The Pulitzer, I trimmed it already. So the beginning is finished and the end. It's our final alignment. Yeah, I see it here. It's correct. Okay. The next step, I'm sorry, let's see. It's nearly 9 o'clock. I would suggest we just stop here for a second, for 5 minutes. Oh, no, let's say for 10 minutes. We have nearly an hour now before we then go on and then really concatenate these 3 genes. This is not so easy. We have to think about several pitfalls, and then maybe you are refreshed. Okay, let's see again, 10 minutes past 9, okay? I will stop also the recording now, And maybe you remind me to go on with to proceed then with the recording. Okay. Good. See you then in 10 minutes.
Okay, thank you, thank you. Mermedi.
Aman di lantai 2.
Aman-aman. Ya hilang apa ini kalau tuh koneksi so wifi mesti itu telat mesti saya connect ulang. Ya automatically putus.
Take a nap dulu ini.
Ya benar Billy.
Take a nap Kang. Recording so pause.
Sudah.
Ya.
Ini boleh Cik Francine pakai Cik Francine Pedata boleh pakai ini. Dia enggak pakai Mega. Apa tadi Kak?
BioEdit.
BioEdit iyo.
Edit. Semua boleh. Kalau ada sequencing boleh karena mau sandingkan dengan itu data-data yang orang saya masuk tuh. Cuma DP coba yang orang mau belajar ketua editing itu karena pakai mata mau lihat itu mana tuh ada ya DP istilah rekonstruksi kota itu kita baru.
Oh itu rekonstruksi Kang.
Itu tuh rekonstruksi jadi dari original kemudian dia panggil data itu kemudian dia edit di BioEdit ya itu DP istilah rekonstruksi katanya.
Baru sudah itu baru di aligning.
Itu yang dia bekerja di BioEdit itu. Jadi bekerja di file, orang mau move up, move down, saya pindah, mau delete. Nah itu kok so mesti keterampilan khusus itu karena dia bilang so gunakan, so nak pakai software, kurang gunakan mata. Ada lagi kote Encik Stella di sini. Encik Stella Kaunang kote ada. Baru udah keluar matahari ini dapat lihat.
Kamu nih di sana?
Ayo baru keluar ini. Ini jam berapa ini? Sobat terang dong jam 9 ini.
Bagus kalau Sis kabatanya pakai bahasa Prancis lah ini Kang. Jadi rupanya mengerti sedikit kita dong. Nanti kalau dia Matt dia mau datang Matt. Heike mau ada rencana mau ke Indonesia tuh.
Tahun depan kan?
Tahun depan ke iya bisa terang kumpul kong.
Mesti siapkan dari FPIK memang khusus untuk ambil ada data analisis apa ini yang DNA begini. Dia bilang dia siap untuk orang mesti siapkan.
Kan kalau cuma ambil data kok kalau di orang Labs tuh boleh sampai mau ada tidaknya DNA terus kirim ke dia atau bagaimana?
Kalau orang lagi kirim sampel kan ke Korea itu.
Oh iya untuk
Sequence no.
Sequence.
Kalau kemarin kita tiba di Bonn, sampai siang jam 2 siang kita dengan Menerbedi langsung masuk di museum langsung kerja dengan BioEdit ini, dia kasih contoh. Karena kita pakai sampel serangga itu kan DP sequence so ada.
Oh begitu ya.
Iya, tinggal ini kita ada bawa sampel itu kan dewasa dengan dia punya stadium awal dong, masih apa DP nama, belum dewasa. Nah, itu yang kita ada bawa.
Oh.
Kita tanya kemarin kenapa kita mesti bawa dewasa yang bawa DP. Dia tertawa, no. Kita tahu karena orang mau lihat tuh bagaimana dari fase kata dia masih bentuk fase awal dan setelah dia dewasa itu yang dia mau lihat dong apa.
Ada perubahan DNA dong.
Itu di kerja yang dorong mau lihat dong berdasarkan tuh konstruksi yang ada ambil dari NCBI kong dia masuk dia panggil masuk di apa sana BioEdit kan itu.
Data
Termasuk
5 menit kita tadi.
oh begitu ya. Bener Nixon ada?
Bagusnya kok dia ada di sini, Marta ada no.
Akhirnya apa? Kau apa?
Bebas ada pertanyaan ya.
Jangan begitulah. Nyanak ner.
Orang mau pergi ini, iya toh.
Di mana?
Pergi Jerman.
Sudah aku Prof sewakili kan kau Prof.
Tahun depan tuh ke grup dah. Tanya anak di Prancis tuh orang.
Iya, titip coklat jom ner nih.
Coklat, iya met. Pertanyaan sebetulnya sebanyak kata, jadi titip coklat. Nanti Ibu Medi yang berbagi itu coklat.
Mantap. Ada kunci Stella.
Iya.
Aman ner.
Aman.
Aman.
Seboleh bagi sekarang itu coklat.
Sudah su pesan.
Masa pesan sudah aman.
Jalan dengan Nerjem sana, pi cari itu dia.
Aduh, Ner James, mau dua pi cari itu dia. Kalau Cik Francis masih di Jakarta ini.
Di Jakarta.
Iya, ada deh anak di sana.
Francis, oh.
Iya. Cik Francis pe anak di Jakarta. Sebenarnya kita kalau nak pulang tanggal 4, tak ada sehingga cari panci.
Makanya tuh DP apa? DP signal lari-lari, tadi setengah mati masuk.
Iya.
Karena menarik, mesti ikuti. Jadi semua ada.
Kasih rekam di live on YouTube. Nah ini ada rekaman, Cik. Orang boleh putar lagi.
Bagus, bagus. Oh iya bagus, no. Supaya orang sudah pelayanan DNA.
Yang rekam Cik tahu siapa? Triksi.
Oh Triksi. Oh praise the Lord.
Heike putus signal dan ini. Kita lihat putus signal di tengah jalan. Ini tanya mengerti.
Nah, Cire ini udah tidur. Ada istirahat ini.
Sama dengan kita data son.
Ini
Hilang
hampir
signal atau?
Ya, beliau ada istirahat kan. Habis 1 jam dia bicara, istirahat dulu. Beliau bicara.
Oh
begitu.
begitu.
Rest for 5 or 10 minutes.
Oke. Bagus ketuk tadi.
Iyo.
Refreshing soalnya so lama.
So lama. Remember when we were students?
Tadi Reni so boleh bikin artikel DNA, iya DNA. Naik pangkat.
Udah pakai
Cik Francine P. Skripsi, tesis itu disertasi.
Itu cuma penunjang for identifikasi.
Dari situ dia mulai sampai baking DP family tree.
Ya itu itu tuh penting.
Kalau pernah baking dan Renzo pasti cerita ini bisa mengerti.
Kita bilang
mengerti. Nanti mau aku tanya ulang nih, biar Billy.
Iya.
Orang lain.
Mener James ini baru sampai kok, Mang. Toh?
Sudah.
Masih jet lag ini. So Winter Wonderland.
Langsung di museum kemarin, jadi mesti supaya
gitu dapat lihat Pak Fresh ini.
iyo APP.
Padahal tanggal 3 orang di Jakarta, Ren. Dia langsung berangkat.
Wah gitu
Iya.
Siapa yang Nerbilita tinggal dan kereta.
Memang tukang bagara sekali.
Siap-siap mau buku Iko, terus daftar.
Dia kata kok PAIM lain. Iya toh? Mickey Mouse dia PAIM.
So Rainy is making fun of you, James.
Hi Heike, how are you?
Most likely.
Hi Rainy. I am listening. I try to understand. I got jet lag. No, James has no jet lag. James is working hard.
He looks fresh though.
No time for jet lag.
Yes.
That's good. That's good. Keep him going.
Okay, shall we go on?
Yes, please.
Okay.
There we go. Okay.
Yes, please.
First, I try to get the this thing running again. Let's see. Can you see it?
You need to on the record, Heike.
Yeah, that's true.
Yeah.
Tunggu.
Oh, wait a minute.
Christine.
I will on first.
Recorded is now going on. Can you see these two alignments now? The three alignments? What do you see?
Yes, we can see. We can see.
That's good. Yep. So now we have really to concatenate this. So when we include more than one gene, so when we have two genes, we will say actually we concatenate these two genes. And in this case, we want to concatenate actually three genes. We have here the H3, we have here the 16S, and here's the CO1. Here the CO1, I cut off all the names, but here not, because I want to show now some problems that usually occur when you work with concatenated data sets. And you have to consider this already before you actually combine or download also the sequences from NCBI. So we have here H3, this is the nuclear gene. And we can see that when we compare it with the 16S, the available data, the overlap of the data is really low. For example, we have here in the H3 Brasiliensis, but here in the 16S, we have no Tambia Brasiliensis. So how should a program tell when you use these two genes? How can a program, a phylogenetic reconstruction program, tell where the Brasiliensis comes along when there is no overlap with the 16S? Same also with Criula, present in H3, not present in 16S. The only overlapping species here at the moment is Tambia marbellensis. We have one sequence here. This is the sequence, and we have one sequence here. And we have Olivaria, that's one sequence H3 and one sequence there. But all the other ones that I put down here for Brasiliensis, Criula, Cava, Stegosauriformis, Victoriasul, there is no overlap. So we don't have both sequences, only H3 or 16S, and that's not so good. And of course, the same problem we have in CO1, you see already, we have a few sequences here, and here are many more. So CO1, we have much more information. And that is, of course, not so good. Okay, now I have the same problem as usual. I cannot go on. It's frozen. Ah, now it's coming. So the pitfalls and the problems are actually is that you actually need all 3 data sets from the same specimen. It's not that you only say, oh yeah, this is a Thambia mabayensis. I have here a Thambia mabayensis. It should also be from the same specimen, not only same species, but also same specimen. And this then already is very tricky to find out. And that's also difficult to retrieve from NCBI. The accession numbers cannot be used here because, as I said, we have here, for example, the mabayensis. It has this 599. And here the mabayensis has 505. So it's completely different. So you cannot also only search for the accession numbers. It's much more tricky. And as you have seen here, very often it's not available for all the species that you wanted. Nevertheless, it is done, it is used very often, it should be used, but it costs a lot of time. So the problem is it costs a lot of time. So what we can use is that the sequences of the different genes have to have the same identifier or the same name. And what you also have to keep in mind that the accession number refers to only one sequences, to only one sequence and not all sequences from one animal. Yeah, this is actually that you really should keep in mind. So how can you do this? Then there are several options. So you can go, for example, if you want to have a certain, like here, a Thambia Zulu. We have here 2 Thambia Zulu. Now you want to know, is there really no 16S and no CO1? And for this, you can go in NCBI again. You click on this sequence that you have, and then you get all information. I have shown this already to you last week. You have then Thambia Zulu, and it tells you who published actually and who uploaded the sequence. So in this case, it was Pola, Padula, Gosliner, and Cervera. This is the publication. You can download the publication usually from the internet. You can have access to this, and then you can also search through this literature, and there should be a table somewhere in the printer or in the supplementary information. Nowadays, very often in the supplementary information, there should be a table like this. And this is something you also have to keep in mind when you are working on your specimens. You have to include for each specimen where you collected it. Each specimen should have your own identifier that you decide how it looks like. And then when you upload the sequence, you also tell them this is the CO1 sequence, this is the 16S sequence, this is H3 sequence. And NCBI provides you with these numbers. But in the literature, then you can see, oh yeah, Zulu, do they have Zulu? They have Zulu, actually. They have all sequences for CO1, 16S, H3. You just missed it by downloading from NCBI. This is how to check that you really got all the sequences. So that's a possibility. So you can deliver by actively going into the NCBI and clicking on this, providing this number, and then you get exactly this sequence. So this is a possibility where you can ensure that you have all the information for all 3 genes. So you can search specifically in NCBI by using the name or accession numbers and store then the sequences in separate gene files. But what is then also very important, you have to use a specific name for each specimen in all 3 data sets. That means you cannot say in CO1, you just use, let's say 1T brasiliensis, and in the 16S file, you just say T brasiliensis, or you have to find a name that is really unique then for the concatenated data set for these sequences. So this is also very important to think about. Also, you can search in your own saved gene files and rename those. So in the edit in the text file and rename them and give them then a proper name. But don't call them Thambia brasiliensis, because when you have it like this, call them, then all the programs will have problems. The names have to differ in some way. And even if it's just you put 1, 2, 3, 4, 5 in front of it. If you want also to search in a different way is, for example, I told you now you can use these ones and then give them proper names, but you can also search for the vouchers. So I see here in the publication, they have these CASIs 78792. So I can go in NCBI and search just for this one. So I put this number here in NCBI search mode, and what I get are 3 items, and that's actually the histone 3. This is the cytochrome, the CO1 sequence, and this is the 16S. By the way, that's why I didn't get it. I used 16S, and here nothing is said about 16S. It's only called small subunit ribosomal RNA gene. And by the way, this is wrong. The 16S is not the small, it's the large subunit of the ribosomal RNA. So you see also these mistakes are still in NCBI and might hinder your search for these, for the correct sequences. Yeah, again, it's not always that easy, but at least when you have seen in the literature, oh yeah, you have these vouchers and you want to have then all the hits, you can also do it in this way. But of course, this is only a small amount of the really available sequences that can be downloaded. Now, when we are going really to concatenate, it's very important, as I said, that the names in the different data sets are the same. So we have here the Tambia CO1, we have here Tambia 16S, and now you have to start to edit. You can do this in BioEdit, as I've shown you, but you can also do it in the editor. I prefer actually the editor. For me, it's much easier, because I have this on the same screen. So here, for example, I renamed this already, this sequence in 1, T for Tambia, Eliora is the species name. I did this already. I actually erased the rest, because if you keep this in there in some way it's united, then this name would differ from this name. So I erased everything and have here only 1 Teliora 2. Yeah, so I have 1 Teliora here, 1 Teliora, so Tambia Eliora here, CO1 16S. Here is the 2. I have a second one. I already called it 2 T Eliora, and this can also have to go away if I don't want to get mixed up. But be sure that you don't lose this information later on also with the accession number, because the accession number has to be mentioned in your publication for this specific sequence. So when you have done this, you have checked your CO1, you have checked your 16S, you have renamed things, you have renamed also the histone 3. Yeah, you do the same there. And then you save it again as FASTA. And now we have Tambia CO1 aligned, Tambia H3 aligned, Tambia 16S. So first we had it as editor, but then you can transform it in FASTA by saving it as FASTA, or you go in BioEdit and save it as FASTA. Okay, now we have these 3 files with the 3 different genes, and BioEdit does not concatenate. I have not found, it's still not in there as far as I know, where you concatenate. That means where you just attach the different genes to behind each other. And for this, now I want to introduce Mega a little bit. I know that some of you have worked already with Mega a little bit. I don't like it very much, but it has a lot of issues that can be used. Mega, the huge disadvantage of Mega is it uses its own format, and you have to transfer actually the FASTA formats into Mega formats. Well, you don't have to transfer it, but later on it will save everything in a Mega format, and then you have to convert it again into FASTA. So it works with FASTA, but it does not save in FASTA. So Mega is also free software. You can download it from the internet, called Molecular Evolutionary Genetics Analysis. That's what's the name for. And that's where you can get the software from. you can open it then, and then you have a headline like this, and there are many options that can be used and that are not so good in BioEdit. They are better here. The first of this is very easy. You open it. So remember, you have the files in a separate folder. I called it actually from Tambia project, Tambia 3 genes concatenate, and then I say 4 concatenate and align. You can do whatever you want, but do it in a way that you find things again. So you click on this one here, TA. This means actually thymine and adenine. These are the symbols for the nucleotides, and that means your aligned files. Then you say concatenate sequence alignments. You click on this one, and then when you click on it, then opens your Explorer. The Explorer opens. Then you click on this concatenate and line, the file where you have the 3 files in there that you want to concatenate. And now you have already one of the first pitfalls in Mega. You don't see here anything because these are faster files and it will not show this, but they are of course there. So you just say, because you know that they are there, you just say, okay, search for, click on the select folder. So select on the folder, and then it will ask you, do you have protein sequences or nucleotide sequences? And then you click on nucleotide, because we are not working on the protein level, we are working on the nucleotide level. You click on that, okay, and then this appears. So this might look familiar for some of you who have worked with Mega. Now the alignment is a little bit differently colored. You have where you have differences, you see actually the nucleotide, this cytosine, where you have the same as in the first line. The first line is the reference sequence here. Then you have just a dot. And then you see already here also differences with colors and also with the dots or non-dots. So this actually is represented now with this one. And you see we have now many more sequences as we started with. Yeah, of course, because we have several that we have only CO1 available, several we have 2, and several we have 3 available. So when we go and look at a little bit more closer, then we can see we also have a lot of gaps. We have also a lot of missing data, and that these missing data are actually represented by missing genes. So in this case, I think we are in the CO1. So we have a few. No, that's not true. We have this. No, this is histone 3. So we have no information for these groups, Polizera and Tambia. There's no histone 3 available. So the program Mega will include question marks, because it does not know how. When we would have the histone 3 sequences, what would there be? Would there be an A, or T, or whatever? So this information is different from the gaps. A gap means that because of the alignment, they had to shift this sequence to a certain area, and actually we are missing the information here. So this is also, of course, might be because we have not sequenced this properly, but it could also be that there is nothing, because there is a long gap. Really, there are no nucleotides in the original data set. So this is something we don't know, but gaps are not the same as these sequences. That's these quotation marks. So please keep this in mind. Now we want to, because I said Mega uses a format that is not used and not acknowledged by a lot of phylogenetic programs. So what you have to do is you have to export your data now in FASTA again. Of course, it's nice that Mega has included this so easily. So you export your data, you click on that, and then you say here, this opens, you give this a title, you say the description, it tells concatenated files. It is by default Mega, but you don't want to have it Mega now, because this is what you can't work with on. So you choose then FASTA, and then you click on OK. And then you have it as a faster file and you can save it. So you have export, that's what I say here, export file is faster, and save again under a good name where you can actually remember what you did. And I say now 3G, that means 3, it's not what we say in the vaccinated and recovered or tested. That's our 3G rule. That's not the truth. This is the 3 genes that we are talking about. Okay, now how does it look like? Now, I use here now the editor again to open this faster file. And I start here with this 1 Tambia Iliora. I use the name 1 Tiliora. And the first one here is the CO1. This is the H3. It's missing. We had no data for that. And this is our 16S. So this is missing. We have, well, we did not have the gene available. And here we have the gaps. And the gaps means that something that in the 16S gene, this species does not have any nucleotides, whereas that's the same for this species, Phantasmalis, but there seem to be species where there are nucleotides. But this species does not have that in this loop region. It's the loop region again, and therefore there are gaps. You might remember that Polycera, the Polycera species had in the loop areas many more nucleotides, and therefore in the Tambia species, we had these gaps. Yeah, so what do we do here now? We have here the CO1, histone 3 is missing, 16S, same here, same. But then you can also look, maybe you have made mistakes, and this always happens. I always have to say, even if you are very careful, you always make mistakes. So in this case, what did I do? I had the same specimen for CO1 and also for histone 3, but I used different names when I was renaming. I used here 3T and the dash brasiliensis for the CO1, but I used for the H3, 3T brasiliensis. And then, of course, the program considers this as a completely separate specimen or species, whatever. It considers this as separate and will treat it also as separate ones because it has different names. So you can go back in your original BioEdit version or Mega version and redo this, but you can also do this here by editing this sheet because this is a faster file and you can save it again as faster file and then have also it corrected. So what can you do actually? You can combine by copy and paste. So what I did here in this case is I can copy this CO1 and include it in this one here. Yeah, I erase the quotation marks or I overwrite the quotation marks with these not quotation marks, question marks. Sorry, I delete this or I overwrite these question marks with this file up here. So copy paste and put it in here. I don't do it the other way around. I don't copy this and put this in here because it's much more difficult to find where is the end of my H3 here, because somewhere here the 16S starts. Yeah, you see here the 16S starts. It's much more complicated. So think in the beginning how you do it. So you can just copy this and put this in here. And then you delete, of course, then this part because now you have this information in there and it's then renamed. And I did it for some stuff. And now we can have a look at the alignment, only a small piece here. We have here the partial CO1, the mitochondrial gene. You remember it's about 650 base pairs long. So from here on, we have the CO1. From between 660 and we will see where it ends. We have the histone 3. So we have the histone 3 information for these ones here, CO1 and histone 3. So we have some overlap here, but there's no overlap at all for these ones between CO1 and 16S, and no overlap also for these ones. So it's a really not so nice alignment. When we go further on. Now we are talking about positions 980. We are here at the end of the histone 3, the nuclear gene, and this now starts with the 16S. And you see there's also a little bit of overlap with the H3, but also not so much. And we have huge gaps in this case, and down here, more information about 16S. So you see, it's a huge gappy alignment, and that's not so nice. So what you can do is also you rearrange to see better where do you have information in these alignments. You go back to your BioEdit sequences, the original gene, the gene alignments, and go and see what do you actually have, and you can then reduce your data set, because there might still be some overlap and you missed it. And it's then much more easy when you arrange things in an alphabetical order. So here in this case, I start with Brasiliensis. That's the first ones. Then we have C, Crioula. Then we have the next one is already K, Cava, Mabeiensis, and Olivaria, O, and so and S, and it starts with Z. And then you do this the same, and then you can start, do I still have some overlap, and start to rename. But always be careful, it should be still the same specimen, not only species, but the same specimen. And then you can start. So I did it here. I checked, this sequence is actually the same as this one. I have overlooked it, because when you have many, many data opening in a small screen, you overlook things. So go back again, check again, and you do it in this way. So you rename this, yeah, you erase this accession number, you call it in the same way as here, Amakusana. You have then here, you rename this, in this case, you say T, Amakusana, don't forget the dash down below there. You apply and close, and then you have a new name there. So this is now a better overlap now already between, this was, wait a minute, this was the H3, this was the CO1, the 16S. So you have already 16S and another CO1 overlapping. And you go on like this. You realize that there are much more overlapping between Brasiliensis and the CO1 Brasiliensis. So I renamed again 1, 2, 3, 4, 4. I renamed here already, and I renamed these ones. They are really the same specimen. One is the same specimen as here. So you see this here. This is another Tambia Brasiliensis. This is the voucher specimen. It ends with 243. You go see here, different accession number, yeah, different gene. It's the same species, and it's also the same specimen. Here you see 243. It's exactly the same voucher specimen. So you rename it again and call it then, I called it 5T Brasiliensis. And so you can match much more to get a much better overlap. Then you save them again, these alignments as faster, as I said. Now I put another name on it just to remember. This is now BioEdit, faster BioEdit. And what is also now, what I want to tell you here is the way how you arrange the sequences for Mega is how it adds things. So in this case, now I rearrange this by putting 1, 2, 3, that it is really in this order, because it's usually arranging by alphabetical order. Then it would start with C, it would put an H and then 16, or 16 in the beginning and then CH. But here I want to have the mitochondrial genes first, and then the nucleus, the nuclear gene. And therefore I put 1, 2, 3. Then I repeat with Mega. I opened this actually, I said concatenate, and then it concatenated. And now you see that it actually concatenated. We still have no information for 16S, it was CO1. We have here the H3, the 5 Brasiliensis. Actually, we have 6 Brasiliensis where we have overlapping data. You can't see it here, but the CO1 is now in the beginning. But it's still, you can see a lot of question marks here. And this is unfortunately a situation we have to deal with. And next week, when we are really then doing the phylogenetic analysis, we will talk about where might be the pitfalls when you have what we call a gappy alignment like this, how this might influence also your tree. So yeah, thank you very much. That's
Okay. Thank you so much.
Shall I stop the recording or wait for the questions?
It will okay. The recording, because the questions also
Okay,
record
yes.
on YouTube.
Good.
Thank you, Professor Heike.
You're welcome.
Thank you so much. So, folks, do you have any other questions for this this webinar, this topic? Encik Francine, maybe?
Pas dulu.
Oh, pas dulu. Okay, but by the way, we already posted in the chat. Would you please fill it for the presence and absence?
Yeah, presence and absence of data.
Absence of data.
Well, I know that this is really complex stuff, and I know also, and I was talking about this with James and Mehdi yesterday too. They are here in Germany at the moment. That for you it is really difficult to learn this in a way that you do this so easily, yeah, and also can teach this very easily to your students because it needs training. I also have to look up certain things again and again, because then maybe it's 2 years ago that I did it. And you cannot keep everything in your mind. And the other problem is additionally is the programs change every 2 years. And then suddenly a program is not working anymore. And suddenly you have a new program with a new format, and it's also not working in the other programs. And then you need programs to convert things. That's why I hate Mega. If Mega would use faster format, it would be used much more often. But because Mega has nice features, but yeah, it's because of the format. And
yeah, Mega keeps changing. Mega 11 or 12 right now.
And therefore, I always say my brain is old. Yeah, I will never learn genomics. Impossible. The pipelines we are using at the moment in your museum, the PhD students, they learn, they are trained. They are the ones who can hand over to the students, to the next generation. I can only tell them about the methodology and what happens within when you analyze a genome. What do you need for this? But the programs, they are so sophisticated now, I cannot teach this. I can tell what you have to look if you're working on genomics. What do you have to look for? Yeah, where are problems, pitfalls? They are always the same. The alignment is the most problematic part. And no contamination in there. Mistakes in naming. It's the same problem you have in genomics when you rename things, or then the programs have problems. Yeah. I know that from in our museum, it happened all the time.
All the time.
So there are pitfalls in the genetics. What I'm doing is genetics. I'm good at that. But in genomics, you have similar pitfalls. So when you know in genetics, you can also say that in genomics, we have to look at that. But using the programs, no way. No way. So I don't blame any one of you when you were not trained or have not really worked continuously on this question. You cannot know. You cannot know. I have worked on phylogenetics now for 30 years. I started, 25 years. I started in 95. And so
wow.
I did my first sequences by myself. And then I had a PhD. I did this together with a PhD. And then I stopped. My PhD took over, and then I never, ever went back in the lab. That's why I say I cannot teach a student in the lab how to do it, because things have changed completely in the last 25 years. And this is why, if you really want, in your faculty, want to work on this, you need a person who is continuously working on the subject and is training him or herself all the time with these programs because they change. So if you decide, like Francine maybe, you decide you want to work on it, yeah, but you are working on conservation, you are doing teaching, you are in management, you cannot do this anymore. You need a person who's only doing this and helping you again and again. You know the background, you know how it should work, but with the programs being updated, someone has to help you.
Yeah.
Yeah.
But anyway, it's so interesting, ma'am, to study the phylogenetic because it's explaining about the population genetic and the speciation itself.
Yeah, this is true. It helps you, but it takes also, and Mehdi and James have seen this already, especially Mehdi, who has also worked with the raw sequences. It takes a long time. And usually, so that's why I said, I am not working with the raw sequences. This is, a student has the time, a PhD student has 3 years. A PhD student can work on a 100 or 200 raw sequences. Mehdi knows now how this means, going through each nucleotide, comparing, is this correct, make decisions, change things, and that takes time because you have 600 positions, and that's only CO1. And then you have 450 positions for the 16S you have to check, and that's hours of work. And that's then only one consensus sequence of one CO1. And now we are talking about population genetics. Population, you need to talk about populations. You have to have at least 5 to 10 sequences from the different localities. If you have only from one locality and try to find out something, you can only say, yeah, intraspecific genetic difference, not populations. Population means from different localities. So population genetics, you should have at least 30 sequences from different localities. And 30 sequences, CO1, well,
it could be in the province or in other countries.
Also including other countries. How is Australia differing in the mutation rate from North Sulawesi? How is South Sulawesi, North Sulawesi, maybe Japan, Philippines, Australia? Of course, you can limit this to North Sulawesi and compare. We have done it. Compare Bunaken with Lembeh, not Lembeh, so we compared Bunaken actually then with Bangka Island, and there are already differences. Must have been something to do with the streams. But we also included the information to say so. We had information from Australia and Philippines. When you do not have any information from other areas, how can you know whether your Bunaken population is actually the same in Japan?
Japan, yeah.
Yeah. So,
and that's what you want to explain about the phylogenetic tree. That will be clustering in what area or in what species.
Yeah. That's why you need. That's why you do not need only your own sequences. You need all information available from out there. But this is already the second step. When you collect specimens, you first have to identify them. And for this, you just need a phylogenetic tree. In this phylogenetic tree, you include all information, all published information available from this group you are working on. So maybe, James, can I use your example? Is this okay?
Yes.
Yeah. So yesterday we started with a cicad, you call it cicad, cicadide. It's from the family Isidae. Yeah. James did not know the genus name, no species name, nothing. But we know it's a cicad. And that's so you have to download actually all sequences of the Isidae. He knows it's an Isidae, that's what for sure. But which genus, which species?
Yeah.
So to make it properly, you have to download all sequences from the Isidae. Not only, well, he has a CO1 sequence, so you have to download all the CO1 sequences from the Isidae. And yesterday we did it, and we ended up with 200 and 80 sequences, I think.
Wow.
Yeah. But then it started already with the first mistakes I made. Sorry, not James and Midi. They didn't know. Because it's CO1 is not working as a keyword, searching for it. So we used, as I usually do, cytochrome. And then cytochrome. But I'm not an insect person. So in sea slugs, cytochrome is only CO1.
Oh.
In the insect people, they also use cytochrome B. Cytochrome B is a completely different gene. You cannot mix CO1 with cytochrome B. So in the beginning, well, we made a quick and dirty alignment, and then I thought, wow, this is so different. There was a very nice group, they aligned properly, and another group that was just different. And then I thought maybe it's reversed, yeah, it's wrong alignment. So we reversed it, we did a complement, we again aligned it again with MAFT, was always wrong. And then I said, we have to look at the names again, what is there? And then I realized we mixed cytochrome B with CO1. So we went, well, normally you would go back in NCBI, you would then write ICIDE, and then you would write cytochrome and also include CO1. And then you would go cytochrome CO1, you would then get much less sequences because the cytochrome B would not be on that place. We did it the other way around. We just had already the alignment. We just erased from in BioEdit, we just erased the sequences where it was set cytochrome B. And then we ended up with 150 and so, yeah. But these are all pitfalls. And I'm not working on the I'm not working on the thickets. And when you're not working on the groups, the less you know, the more difficult it becomes. And the more you work with it, the easier it will be. So I think, although James is working on the ICIDE, he's not trained in doing this sequencing stuff, he would not have found out probably, yeah? So what the mistake that you can do is then you have the alignment, you see, oh, this is a very nicely aligned group, and this group is just different. You make a phylogenetic tree, and you end up with one group that is then monophyletic, and the second group is also monophyletic. And so you say, oh, nice, yeah, this is the ICIDE, they belong together, and you say, oh, and they belong together. Oh, no. This is because you use different sequences in there, and you cannot say anything about phylogenies. But when you don't know, and then publish, this would not be good. And for this is, when you are not training yourself again and again and again, this is difficult. And that's why it's good to have a person who knows about this and can help you when you have things like that, when you realize something is wrong in there, but you can't find out. Yeah. Billy, you are muted.
Maybe Dr. Rainy, do you have questions or something you want to say?
Very, very interesting though. After being a while away from that work, it's actually a refreshing thing. But
Yeah.
as you said, the program keeps changing, so we have to actually keep working on it. Otherwise, we're left behind, right?
Yeah.
Thank you.
Okay, Siska, Professor Siska, maybe you can ask questions.
Boleh, Bill.
Okay, silakan, go ahead.
I want to ask you, Heike, about the first step for filtering to submit, like COX-1. That's the important to find out the data.
Yeah. If you want, I can just open the NCBI file and then we can discuss it, because I think we still have a few minutes, and we can do this then online and you can just ask. I only have to open this first. Go here in NCBI. Yep. So I share my screen again. Yep.
Oh.
Do you see it?
Yes.
Yes, we can
Yeah.
see.
Okay. So when you go in there, you want to only have the information for the nucleotides. You want to have the sequences. So don't forget to click. I made the mistakes yesterday again. Click on the nucleotide. So now you're only searching for the nucleotides. Okay. Shall we go for the ICIDE again? Yeah,
Okay,
let's
okay.
put ICIDE. That was these cycles James wants to work on. Yeah. Do you see ICIDE in the headline?
Yes,
Yes,
we can see.
Okay, that's good. Now I search. Okay. So you see here it opens 843 sequences. And then you see already there probably, yeah, here already in the first ones here, you see ICIDE. It's probably Oisudazina, but they don't know. So that affin means affinis, it's close to, yeah, it's probably, but they don't know. So this group seems to be very difficult. And you see here, this is 16S. This is the small subunit of the nucleus. Then you have here 28S. This is the large unit. If you want to have a really large concatenated data set, you could then, oh, no. You could not use, you cannot concatenate these two ones because this is a different species. Yeah, so no option at all. But we have 843 sequences in there. If you go down, 28S, 28S, 18S. So it would probably, yeah. We have here already cytochrome oxidase, subunit CO1. So in this case, if we go now for search cytochrome, no, if we go for CO1, then we will see how many we will find. Yeah. So there are cytochrome oxidase subunits CO1 in there. Well, we know we did this yesterday. So let's go for ICIDE CO1. Well, it's actually COI and not, let's go for CO1 first. You see, no items are found. Okay. Now let's go for COI.
No item. COI.
Yeah, and now COI 133. So James, if we would have done rightly CO1 yesterday, we would have ended up with 133. Now let's, because we ended up yesterday by deleting the cytochrome B, we ended up with 150, I remember. So we are still missing a few here. So let's try to get them. Now I put in cytochrome, just cytochrome. Now we have 284. Many more. And here already the first one is cytochrome B. So the abbreviation is cyt B. This is, to my knowledge, no barcode gene for identification. But I am, as I said, I'm not a specialist on this group. So if you want to know if this is used for barcoding actually your species, you have to know the literature. And in the literature, it will tell you, what do you find more, cytochrome B or CO1? I don't know at the moment. What we can do now, we have 284. Let's check. Do we have more CO1 or do we have more cytochrome B? CO1 was 133, I think. Now let's go for cyt B. $74. It's still a lot. Yeah, so probably it's also used for some species identification or for some phylogenetic analysis. However, when you, James, you have already a CO1 sequence, so the cytochrome B does not help you at all. You can actually forget it. Yeah. If you want to work on the phylogeny in a way, you could also download cytochrome B, concatenate it with CO1, have your sequence as CO1, and hopefully the CO1 of your sequence has enough information to attach it to the correct CO1 sequences of the group you want, where it is in there. And the cytochrome B helps you also then in the phylogeny, who is more related with what. Yeah, that would be a way how to do it. But as I said, cytochrome, let's put it cytochrome. How can we find the CO1? Because we had more CO1s yesterday than cytochrome. When we go for CO1 now, let's see, still 133, does not help. So with this CO1, we will miss something, because maybe they also, what could they also use for that? Cytochrome subunit. Let's try subunit 1. Let's try subunit 1. Subunit 1. 220.
Oh, 220.
Okay, so you see that this might probably be the best one. But what I would always do now is, when we don't click on specific ones, when we just keep it like this, what you do is next is send to. We have complete record. We use file. We create faster, and we create the file. Where is it now? I have to. Oh, it's not, it did not work. I don't know why. It did not work. Ah, no, sorry. It's here. I open it. Okay. Do you see now the editor file, or do you still see the NCBI? Or do you
Still NCBI.
Okay, so I have to close this one just to show you. Okay.
Yeah.
This is now, this is now the file, how it opens. So there should be, this should be a huge file. Yeah, you see, wow, lots. But then you can also look for, oops. And I see here, for example, we were looking for the subunit 1, but we have here also the large subunit ribosomal RNA. So I see here already, when you use the word, we used subunit 1, it's not working. Because we have here a mixture again with, this is large subunit ribosomal. This is a ribosomal gene mixed with a CO1 gene here. So especially with this cytochrome, the CO1, it's really difficult to find all of them. So the best way is still using cytochrome and then CO1 or CO1 to find them all. And that it did not work with the subunit 1 is because when we go for here, the ribosomal, there's somewhere a 1. There's also a 1 in here. That's probably why here's a 1. And that's why it's using also this information, because it's not in a row. When we have it separately, it searches independent. It searches independently in this row. So if you would go for partial sequence, it doesn't matter where the partial sequence is mentioned. Or partial, it would just search where is this in there. Yeah. Okay.
Okay. Okay. Somebody have a question? There's a question. Indra. Or James, are you okay?
Yes. Okay.
Okay.
Should
But
we continue at the museum?
Yeah, if it is not clear, you can ask her.
Yeah, but
So,
what I just did at the moment, this is one of the most, not the most, but it's also a very, very important step in your analysis. If you don't know where to find the information that is published, then a reviewer, and when you don't use these data, then a reviewer will always say, wow, there are so and so many published data in the NCBI. Why did you not include this in your study? Yeah.
Yeah.
In an international acknowledged journal, a reviewer will always ask for this. Always.
The latest publication that you have to cite it.
Not only the latest, you have to include the data that are published. And then even in the morphological data, if you are working on the ECDA and you want to now know, is this a new species? You cannot rely only on the molecular data. You also have to look for the old literature 100 years ago. Maybe this cicade is already described by morphology in 1920. 1920, there were no molecular data, but there's information available for this specific species that James brought to the museum here. And the molecular data, they are not everything, yeah? So if you go for population genetics, you don't have to consider so much the morphology. If you're going for a new species and biodiversity, you cannot skip completely the morphology. You have to keep this in mind.
Yeah.
That maybe in a genus, yeah, maybe this belongs now to the genus, I don't know, Tetragonus, whatever. And you have, let's say, 30 described Tetragonus species. In NCBI, you have only 10 species, sequences for 10 species. There are still 20 that are described and they are available, they are valid. And you need to know about them, whether your species you found in the mangroves are belonging to one of these described ones. You cannot say because no molecular data available, new species. This is no way, sorry. So it needs also, if you want to work on these things, you also need some information about the groups. And this is why I say I'm working on sea slugs and I have no idea about the tickets. If you want to really describe species there, you need an expert who knows about this. I don't know about it. I don't know the characters you have to look for, what is important. I can do the molecular part and give you the tree, and even interpretation, whether this is correct, I have no idea. Yeah.
I remember that you say about the first time about the generic species or something.
You mean a speciation or?
Yeah.
Yeah.
So we cannot also rely all the data from the genetic data.
No. You should at least know a little bit and say then in the publication, you can say, okay, from the genetics, my species differs from these other genetically defined species, but you have to write in your publication. However, my species looks also very close from the morphology to the species that was described in 1850 or even 1795 by Linné.
Oh yes, I remember cryptic species you mentioned.
Yeah, and we did it for the Phyllideids, and it's a lot of work. Yeah, and this is why we also train students only in one group. We don't train them in corals, sponges, echinoderms, in all these groups. They decide, I want to be a specialist maybe in flies. I want to be a specialist in sea slugs. I want to be a specialist maybe in a certain plant group or algae. Yeah. Then you can specialize and you learn about the group. You read the literature on a regular basis. You can collect the data, and then, yeah, then you are in the group and you can judge much better. There's something wrong here, or of course there are many described, but no sequences there. It's a lot of work. And this is why a good publication on biodiversity is also not done within a month.
Yes, yes. Indeed.
And this is something you have to tell your ministers, and whenever you have a chance to tell this, tell them, if you want to have high-ranking publications, they need time, because analysis takes time. You have to have the time. If your minister say, no, you get the money in April, I want the publication in December, or even earlier, they actually they hinder you, they keep you low. They keep you low in quality. And that's not your fault, it's their fault. Yeah. Sorry, I'm very direct again.
Why are you doing that, James?
But anyway, we already take 10 minutes more.
Yeah.
However, it's very fruitful conversation,
Yeah.
I guess.
Okay.
Thank you so much, Professor Heike.
You're welcome.
I hope we are all here. Cik Francine, Cik Reni, dan Profesor Siska dan lain-lain, Wiki and etc. We would like to see you in Germany. Hopefully next program we can visit you there.
Oh, Bill, we will have the last talk on 14. Yeah?
14, yeah.
Tuesday. Yeah,
Yes.
Okay.
exactly.
Oh,
So hopefully,
next week.
yes, next week, Tuesday.
Okay.
And then we will
last
talk
meeting.
we will talk
Okay.
about the phylogenetic trees.
And then we will
Oh.
see the
More
phylogenetic
interesting.
trees, yeah?
Yes. Yeah.
Okay.
Because now we have done already the alignment. Yeah, you have the alignment. Now the next step is to get the tree.
Okay.
Okay. So thank you very much for your interest actually and for the nice questions. And yeah, I hope it helps you a little bit. And of course, when you have questions also, yeah, you can ask. You have my email address and you can also ask. You know, I will always help you. I will try to help you.
Thank you so
then.
much, Heike.
You're welcome. Bye-bye. See you
Bye
next time.
Bye.
We close
Thank
this
you.
meeting.
Welcome.
Heike.
And would like to say thank you so much to Professor Heike, Dr. Medi, and Dr. James Paulus.
Dankeschön.
Bitteschön. Sama-sama.
Sama-sama. Bye and all the best. Good luck.
Ya, selamat
for your
tinggal.
research. Selamat tinggal.
Terima kasih, Prof. Heike.
Thank you. Terima
Sama-sama.
kasih,
See you
Prof.
next
Heike.
time. Banyak.
Kasih Menir Franz.
Oke, sama-sama.
Nerjen,
Yes.
Yo, Bon. James Bond.